I had a really hard time deciding what specific topic I wanted to cover. I knew for a fact when we did virology, I wanted to do something related to HIV because it's just a topic that's been really interesting, and especially with my time abroad last summer, it's like very, I don't know, it's like a touching, moving, wonderful subject thing. And it's kind of been like the nice bridge between my interest in like biological science and global health. So today we're going to be talking about the development or the potential of developing a pediatric HIV vaccine. So yeah. Oh no, where's the clicky button? Okay, cool. So these are today's objectives. Ideally, we kind of just want to review. So I stole some of my old notes from my global health and infectious disease course to go over the basics of HIV and how it infects and whatnot. So we're going to kind of understand from a global health perspective how the epidemic kind of came to be. And then from the more biological perspective as well, just like the molecular process of viral entry, infection, and kind of how it interacts with the host immune system and essentially why the host immune system fails. And then we're going to be looking at some really cool developments in pediatric vaccine progress. One of the PIs that I interviewed with for grad school interviews was Christina de Paris, who has some like really awesome research in this field. So throwing that in here. And then we're just gonna like end off talking about the significance of what we can do in terms of global health and biological research today. So we are going to rewind back to the beginning. Like I kind of mentioned, the study of HIV/AIDS is something that I hold really near and dear to my heart for a couple of reasons. So for one, the molecular understanding of the disease only really comes to have meaning when we understand it in the larger context of human health and disease and global health specifically. So the HIV/AIDS epidemic was actually a turning point for the world in that it was kind of the first time that we looked at health and disease from a human rights standpoint. And this is kind of like where we see today with the whole COVID pandemic. People are afraid of this disease and people who have it, and it's kind of become in its own respect like a political weapon, and a lot of that is rooted in the fear and what we don't know about it. And this was kind of like the first time that we saw an example of this when we saw the outbreaks of HIV/AIDS. And just like with COVID, a lot of very ugly things happen when we don't have a concrete or proper understanding of the biological basis of it. We still don't know that for COVID right now. But back in the 1980s, this was kind of when HIV and AIDS first emerged. They were categorized into these four main groups, particularly in the United States. So starting off with the first group, in 1981, doctors in New York and California started to observe an increase in Kaposi sarcoma and pneumocystis carinii pneumonia in gay men. And this raised a lot of red flags for doctors because these were specific diseases that typically were not observed unless a person was immunocompromised. And at that time, they didn't have an understanding of HIV and AIDS. There, of course, it wasn't named at this point. So they just kind of looked at the specific population where they were observing these different symptoms, and they just kind of categorized it in terms of saying, well, this is only a disease of the homosexual population, and that's also kind of where the stigma first arose. So then in late 1981, reports then began to emerge of outbreaks of pneumocystis carinii pneumonia in people who were injecting illegal drugs, so heroin users, for example. And then by the mid 1982, at that point, these same diseases were also identified in Haitians and hemophiliacs. So this is kind of where we started to see this othering in the context of what communities these diseases started to affect. So in addition to just seeing this progression of the demographics that this unknown disease would infect, the CDC didn't really help in that they released a statement about these specific four groups being very high risk for the disease, and even got to a point where there was this one student, his name was Ryan White, and he was a hemophiliac who had actually contracted AIDS from a blood transfusion, and he was barred from school, and this was during the Reagan era, so it was backed and everything at that government level. So that also kind of created more of a divide in terms of with respect to how society viewed it, because not only were they seeing these patterns coming out in the media, but there was also issues in terms of the CDC and government officials like the president who were supporting that kind of dialogue around who and how it could infect people. So obviously the story has a lot more chapters. There's a very long-winded history and backed up movement and everything. But I think the main point that I wanted to make here was that the understanding of viruses goes far beyond just the bound of biological science and medicine, and its implications on society are vast and far-reaching. So we have to recognize where implicit biases exist, even in what we may think is an objective science. Okay, so we're going to start off with some of the HIV basics. So HIV is a lentivirus, and therefore it is characterized in terms of causing long-term chronic disease, so long incubation periods and periods of latency, followed by chronic symptoms. And in this case, we get the progression of that HIV infection to AIDS. So initially, in the first few weeks of infection, HIV presents itself in an influenza-like virus in the blood. So you can see that in this diagram with the red line. So very high viral load at this point. And then in response, obviously, your immune system, specifically your adaptive immune response, kicks in, starts to kind of handle that high viral load. And then these CD4 or these specific T cells, those levels are restored essentially to normal-ish levels in your body. But obviously HIV, as we know, has the possibility to become latent in the body, and it can create this progressive infection that depletes that CD4 positive T cell count over time. And that's when you start to see AIDS, so that late stage disease. And typically people who have that late stage disease die from other infections or whatnot. There we go. Okay. So how exactly does HIV enter the host system and cause an infection? There are a few important components, or in this case, the anatomy of HIV, that we want to mention. So for one, it's an enveloped virus, and it has protein spikes on the outside that mediate its viral entry into host cells. And it also has an RNA genome that has two unique or distinct strands of RNA. And then these are actually the basis for creating that proviral DNA through reverse transcription. And so the enzyme itself, reverse transcriptase, is it has a very low fidelity, it does not have proofreading capacity. So being able to essentially cause several different mutations is also what allows HIV itself to evade the immune system. And then the integrase enzyme is responsible for basically putting in that viral genome into the host genome, and then protease itself is another enzyme that's important, and it's responsible for cleaving out the polyproteins to form these new viral particles. So those are just some important, this is what HIV is and what it is. Okay, so in terms of HIV infection itself, via intimate contact, virions enter, and then they must cross the mucosal and epithelial barriers. And the HIV particles have actually evolved to be sticky to these specific immune cells called dendritic cells. And then these dendritic cells, which are capable of endocytosing things that stick to them, so in this case, HIV, it then breaks down and then presents these pathogens, antigens on their outside. And are therefore then APCs, or antigen presenting cells. And then dendritic cells are then able to migrate into the lymphatic system, where they contact these CD4 positive T cells, which is one of the main kind of like reservoirs for latent HIV in this case of viral infection. So two important things I want to kind of point out, or things I'm going to talk about with HIV here, are these envelope proteins. So we have GP41 and GP120, so these are glycoproteins. And these are essentially the important components that form that binding complex with the CD4 and the co-receptor, which I think is like CXCR5 or something like that. But essentially you have this binding complex formed between these viral spike proteins on the envelope, and then this receptor and co-receptor on the CD4 positive T cells. So GP41, which is like this upper portion, I think you can see the mouse, is responsible for anchoring this tetrad here, which is the GP120. And GP120 consists of the 3 molecules and has these variable loops. So the important kind of things to note about the structure here is that this kind of inner region here is responsible for the functional binding. So it's that universal function that allows kind of like that complex to form, and then the fusion of the envelopes of the T cell and then of the viral envelope. But then it also has these things called the variable loops that are components of that GP120. And because those can take on different variations in different forms, it's very possible for HIV to use those variable loops as a mechanism to evade the immune system, because it can change a lot of times. So we'll kind of talk about that with respect to epitopes in the next couple of slides. But the important thing here is that you essentially want to know that this kind of window dressing is a key way to kind of distract or evade the immune system, so that even if you have mounted previous antibodies or some kind of response, it is still very possible for these parts to evade. The immune system will not basically bond to prevent it from entering, but because they change, it's still possible later on then for this functional unit of the GP120 to allow for viral entry and the fusion of the two membranes. So yeah, so then once you have this important binding step take place, what you see is that the viral genome is able to enter the cell, as you see here, and then reverse transcriptase goes to work to form single-stranded RNA, and then cDNA, and then that enters into the nucleus to be integrated into the host DNA by the integrase enzyme, which I mentioned earlier. And then as far as we know at the moment, the site of actual integration kind of occurs at random. So that's also another factor in terms of seeing the phenotypic differences in what we observe in different people with HIV, depending on the site at which that new genome is being integrated. That can also, like the placement on the host genome, can also affect the phenotypes that we see as a result of the infection. And then each time the cell replicates, obviously, we have the viral genetic information that is being replicated and because it's integrated. And then the packaging to create new viral particles is also going to be mediated by protease. So, oops. HIV itself has a genome of about 10 kilobases, and so it makes the most that it can out of the limited space that it has by transcribing these large polyproteins, which are then spliced down by protease. So if you kind of see here, protease is one of kind of those sequences that come from those larger polyproteins, those set 3. But what it will do is that it will cleave itself out, and then it'll find its specific sites throughout the actual polyproteins, and then cleave those down so that these different components can form like preceding virus particles. So the other important thing to note here is that protease itself can actually be a pretty significant target for stopping progressive infection. So to prevent AIDS, you can essentially work on therapeutics and drugs and what, which they have done, to try to target protease to stop the creation of these new infectious particles. And then in addition to these larger structural proteins, a couple other genes I do want to point out just in terms of HIV's capability to be infectious and to evade the immune system are these two genes that are called the VIF and the NEF, which are, this is NEF and then VIF over here. So NEF itself was discovered from studying long-term non-progressors, so people who were infected with HIV but who would not later on develop AIDS. And NEF is responsible for downregulating the expression of MHC class 1, which is kind of this like marker that occurs on all nucleated cells. And therefore it kind of prevents infected cells from putting out these MHC proteins on their cell surface. And as a result, this means that the infected cells kind of become not visible to T cells. So these T cells are responsible for fighting off the infected cells, cannot differentiate which ones they have to destroy. So it kind of allows for this like hiding capability of the virus itself. And then the other thing I want to note later on, which we'll probably talk about, but VIF itself is also responsible for another immune evasion function in which it will target this kind of like sequence called ApoBec. That was another topic I want to talk about, but it was a whole nother big thing. Anyways, ApoBec is essentially responsible. It's a cytidine deaminase, which means it will take those C's and turn them into U's. And the stop sequence for codons, right, it's a lot of U's. So if you're able to basically cause that mutation where you have a high frequency of those U's, it'll start to target that specific, or those specific molecules and the proteins that are synthesized from it for proteolytic destruction. But HIV has the capacity to kind of use one of its genes, which is the VIF gene, to inhibit the action of that ApoBec cytidine deaminase, so that it will prevent the destruction of some of those viral proteins. So that's just like another important target that we'll talk about. Oops. Okay, so now that we've kind of had a general understanding of HIV, how it infects, and its structure and whatnot, the other important thing to kind of talk about here is why does the immune system itself respond or fail to respond to this infection? And typically it can be attributed to these 5 kind of scenarios. So for one, oops, where is it? There we go. So for one, integration latency, we talked about this briefly, in which having the viral genome become a part of the host genome, it allows it to be undetected. The other one, so the second one, is destruction of the CD4 positive T cells. So again, like we mentioned, directly the MHC due to the NEF prevents that distinction between infective cells that need to be destroyed and the ones that are kind of healthy and able to operate. So by having the distinction, it's able to infect and continuously reduce the number of CD4 positive T cells that are circulating in the host system. Third one is the inaccessible epitopes. So like we talked about earlier, the significance of the GP120 and GP41 regions on the, those spike proteins on the envelope. They are hidden until later on because of those variable loops, that window dressing that kind of evades the immune system. 4, we have antigenic escape. So like I mentioned earlier, RNase H, which is one of the enzymes associated with HIV, is very low fidelity. So by having like literally thousands of mutations occur with each replication event, you are essentially creating new opportunities for it to evade antibodies that have already been developed against versions of the virus that have been synthesized. So you constantly need to have more antibodies that will fit and bind to prevent that viral entry. But it's essentially with high error able to mutate and prevent that immune response. And then like I mentioned earlier, VIF blocks ApoBec, so you cannot target it for proteolytic destruction because you're not getting that mutation from C to U, which would essentially allow it to be destroyed. So these are kind of like the main identifiable reasons for failure of the immune system in response to HIV. Whoops. So this kind of leads us to the question as to why specifically, like why has there been an interest now more recently in looking at developing a pediatric vaccine? And there are several really good reasons for this, both in the context of the biological side of it, as well as kind of more the global health policy rights-based approach to it. So just to kind of give some background, at the moment, there's an estimated 1.8 million new HIV infections every year, and that actually includes more than 150,000 infections among children. And infections in those under 15 years of age are typically due to MCTC or maternal transmission, so from mother to child. And young adults and adolescents account for about 1/3 of the annual HIV infections, which significantly increase risks among young women. And young women actually around the world, especially in the global south, are 3 times more likely to be infected with HIV as opposed to their male counterparts. So as naturally we can see that if we're seeing a high rate of infections in young adolescent women, who then go on to bear children and potentially pass on the HIV to their child, then that's also why we're seeing that these two are inextricably linked. So a few reasons why aside from this global health standpoint of why it's important to address this as a global health issue, from the biological standpoint as well, if we were to develop a actual pediatric vaccine, that would mean that there's a very long window between the first vaccination for an infant and then potentially a later dose at adolescence, which allows the body or that body of the child who was vaccinated a very long period of time to actually start to mount and develop that response before they enter an age where they would essentially debut that risk exposure to sexual contact and activity in which they would contract HIV. So kind of ensuring that window through developing an early stage vaccine, that's something that's been very appealing to researchers. And then the other side of it here is that people who were diagnosed with HIV, at the end of 2019, about 67% of people who were diagnosed had access to antiretroviral treatment. So ideally, if someone who has HIV/AIDS is able to take these ARTs, viral load will be low enough so that, for example, if they were to bear their child and even breastfeed and whatnot, it wouldn't be transmitted. However, with vast inequalities in terms of access to these ARTs and the expense that it would impose on people, especially in the global south, who don't necessarily have the financial resources to obtain these medications, it's also kind of disproportionately affecting them in that sense. So it's the idea that we may have the science available, but it's inaccessible to a large part of the world who essentially need it, and especially those who are in these low and middle income countries where we are still seeing a disproportionately high rate of new infections each year. So despite the fact that HIV treatment and prevention programs have expanded over recent years, there is a really big need for addressing pediatric AIDS, and through a vaccine, that's one really great potential that we can look into. So this is kind of why we want to look into developing this vaccine. So in terms of recent studies, broadly neutralizing antibodies, or BNABs, have not been very successful. However, some other studies that looked at this in animals found that the SOSIP, which is just kind of a recombinant protein that mimics the HIV glycoprotein, so some of those like viral entry structures that we talked about, they actually are responsible for rapidly elucidating these broad and potent antibodies in calves, and then GP120 can also induce broadly neutralizing antibodies in autoimmune mice with breech peripheral tolerance. So a few of these studies were connected to animal studies, specifically looking at these envelope spike proteins. And then the other interesting component was that when you looked at the immune landscape of infants specifically, there were some different regulating factors and a different degree of tolerance in the infant immune system. That kind of raised this interest when you look at like the envelope spike proteins and developing these different combinations of epitopes and broadly neutralizing antibodies. So we also see in infants that there's also less immune programming done by the environment, obviously because they haven't been in the world for too long and developing a response or immune response to their outer surroundings. So this very kind of early blank slate-ish kind of immune landscape is providing a good opportunity for it to say, hey, maybe things that didn't work in terms of developing or eliciting the response of these BNABs isn't necessarily the fact that BNABs aren't themselves effective, but it could be the fact that the immune landscape itself is better fit for a certain window in development. In this case, potentially being pediatrics. So in 2017, the AIDS Vaccine Research Subcommittee, or the AVRS, actually kind of looked over a summary of the literature in the field. And these are just a couple of them. We have a few here from Duke and from UNC, so not bad to be in the research triangle at all. So John E. Sleesman, who's this first guy here, he's from Duke, Dr. Sleesman. His research essentially found that early life is a time in which the immunological landscape is incredibly dynamic, and this could provide an opportune time to induce protective HIV immune responses following vaccination that would last essentially a lifetime. And particularly due to the fact that we have high germinal center B cells, this like high activity, which is, and lower frequency of B10 regulatory cells. We see relatively high levels of somatic hypermutation, or SHM, negative sequences in IgA and IgG in classes in the first year of life. So essentially what his research found was that by looking at the infant immune response and its actual landscape, because these early germinal B cells that essentially select for specific antibodies are more active than the regulatory B10 cells, which kind of restrict some of those host immune responses, that this could be the key to actually developing some of those broadly neutralizing antibodies. And then some of the other studies kind of looked at, so Dr. Hayes, which is this other guy that is beside him, he also looked at these marginal zone B cells, and he found that they're enriched with autoreactive and polyreactive cells. So they're kind of looking at the fact that B cells that are self-reactive may be needed to develop these broadly neutralizing antibodies. So his work kind of looked at the classifications of B cells that they found, with humoral response that they found, and that a specific type at a specific time could be essential for the development of these BNABs. And then, what else was there? Another key study was conducted by Dr. Overbaugh. He's not on this slide, but essentially what they were looking at was the fact that, the level of somatic hypermutation, so the amount of differentiation that occurs with those like the antibodies themselves. So if you have a low level of that somatic hypermutation, it's more likely that you'll have one, more broadly neutralizing, and two, more potently reacting antibodies, as opposed to those that have high levels of somatic hypermutation. And typically what we see in adult patients who have HIV, because they've been infected for several years, and it takes several years for their bodies to elicit an immune response that is able to cover a broad range of epitopes, there are high levels of that hypermutation or somatic hypermutation in these adults, whereas in children or infants that have HIV, because they're actually able to rapidly elicit these neutralizing antibodies, there's a neutralizing effect, and that's pretty potent, but it doesn't have that somatic hypermutation component, or those levels are a lot lower compared to adults. And then two other people I kind of want to talk about is the one woman in the picture here, go feminism, is Dr. Christina de Paris. So this is one of the PIs that I met. She was super cool. And a lot of her work, especially more recently, has kind of looked at the role of adjuvants, or these additional like booster ingredients, in creating vaccines, and how kind of switching out different kinds of adjuvants can boost the actual effectivity of the vaccine itself. So she was saying that the adjuvants that utilize TLRs or toll-like receptors, as opposed to alum or that specific component that they use in adjuvants, have been more effective, especially in that earlier window of development in like pediatric vaccination. How adjuvants might have to be changed across the ages of development. So currently most of the pediatric vaccines utilize alum, but she's suggesting that TLR has been pretty successful in some of her previous studies with non-human primates, and that's something that can actually be used moving forward. Yeah, okay. Cool. Whoops. So kind of a summation of what this conference with all these brilliant minds found was the fact that the first few months of life are incredibly critical for the establishment of immunity. And it's during these early stages of life that we see a much higher level of immunoplasticity. And then this level of immunoplasticity itself can potentially be harnessed to modulate targeted lifelong immunity. So development of breadth of antibodies has been associated with clinical factors such as the time since infection, high virus load, and early decline of circulating CD4 T cells in adults. But when we kind of look at the analysis in infants and children, these broadly neutralizing antibodies are able to be developed in 1 to 2 years of infection, and about 7% of these HIV infected kids they actually remain clinically healthy and maintain a normal for age level of these CD4 positive T cells, even in the absence of antiretroviral treatment. So therefore, HIV infected children have been shown to develop neutralization breadth earlier and at a higher frequency as opposed to their adult counterparts. And this is potentially due to the upkeep of T cells. So the other thing that they looked at when they looked at studies between adults versus children was the fact that we looked at cross-clade broadly neutralizing antibodies. They were detected with children within the first year of life, and non-progressors obviously had a larger neutralization breadth. And there tended to be some variations in these broadly neutralizing antibodies that would target several different kinds of epitopes, and those would include targeting those regions on the variable loops on the GP120 that we discussed earlier that is typically used as an immune evasion technique by the virus. So because in a lot of these children who were infected, they found that those specific antibodies that were developed in response against it were able to neutralize against those variable regions and specific epitopes, then that's also one of the promising findings that they found in infants. So it's really likely that cellular changes leading to developmental convergence occurs, and studies have found that IgG and IgA class switched B cells progressively gain mutations, reaching about 75%, like 75% of those mutations as we see in adults. So it's occurring at a faster rate in children. And the fact that newborn antigen-presenting cells tend to express high levels of IL-6 or interleukin-6, which is responsible for the growth of B cells and antagonistically regulating T cells, as well as interleukin-23, which is responsible for maintaining T helper 17 cells. It kind of shows that there's a skew in the infant immune response toward the humoral response or the B cell response, as opposed to the T cell response, which is also what indicates that the development of these antibodies is one of the key features of the infant immune system. Oops. Okay, so one of the studies that I looked at was specifically looking at the actual action of these broadly neutralizing antibodies in infants and then comparing the effectivity in adults. And like I kind of mentioned earlier, the key difference that we're seeing here across these two studies is somatic hypermutation, or those levels. So adults exhibit a significantly higher level of somatic hypermutation due to the fact that they've been infected for several years and their antibodies have had several years to undergo the process of affinity maturation. So they've changed several times in response to this long latent viral infection and a lot of viral escape. And this occurs several times and goes through several iterations in adults before they're actually able to develop a pretty substantial level of neutralization capacity. Whereas in infants, they're actually capable of developing these broad responses very early in life, and they exhibit, as a result, very low levels of somatic hypermutation, likely due to the fact that they can develop these antibodies significantly faster. So studies have showed that broadly neutralizing antibodies can bind to the virus and that this interaction may actually stimulate germline B cell receptors to initiate the development of broadly neutralizing antibodies. So here in this chart, we're kind of seeing this comparison, the IC50, which is the concentration of inhibitor needed to reduce response for the binding, in this case, by half. So what they did in this study was they looked at a series of different antibodies across the top in response to these different kinds of viruses and their respective tiers and clades on this left-hand panel. And they were trying to see which antibodies were able to neutralize and act against the virus itself. So one specific antibody in infants stood out, and that was this BF520.1. So as you can kind of see here, it was able to inhibit at significantly lower concentrations than these other against these viruses as compared to some of the other antibodies. So in this case, because they had identified the success of this BF520.1, what they want to do is they want to essentially compare how this infant antibody, so it was isolated from an infant, this BF520, would react in adults, which is what we see here. So the infant antibody, as you can see here again, BF520.1, had a greater neutralization breadth than those first generation antibodies in adults, but it wasn't as potent as some of the others. So essentially the experiment kind of showed here that this antibody that had been isolated and identified and characterized from that infant, from a response that had been elucidated, it had a comparable degree of neutralization breadth, but it wasn't as potent, but it was effective considering the fact that it came from an infant. You can essentially put it in against these other strains in an adult and it would still pretty much work, which was pretty exciting. Whoops. So they essentially want to look specifically at the epitope specificity of the specific antibody, and the infant antibody had a similar neutralization profile to another antibody that identified in adults that targeted the specific region called the N332 glycan, so it's a specific binding site, and they found that by adding this N332 glycan, this region here, it would increase the neutralization sensitivity by a 17-fold decrease in the inhibitory concentration. So that essentially told them, or what they found in the study, was that the infant antibody is capable to target a similar site as, or on this envelope region here of the HIV, as some of the other promising antibodies identified in adults had. And that by kind of introducing it to the specific region here with the N332 glycan, it can actually be an important interaction that they might want to look into moving forward with vaccine development. And then we looked at the actual sequence characteristics of the infant versus the adult broadly neutralizing antibodies. The infant antibodies had different heavy chain rearrangements. So the Y-shape, the heavy chain outside was different for each of the infant antibodies themselves. So this also suggests that they were produced by different B-cell lineages, which is kind of correlative with some of the previous literature that found that the infant immune system is kind of more skewed toward a humoral versus a T-cell mediated response. So that also kind of matches some of the previous literature that they found, and that all of the neutralizing antibodies identified in infants had significantly lower levels of the somatic hypermutations as opposed to the adult broadly neutralizing antibody counterparts. So this BF250.1 actually had a significantly lower mutation. So all in all, in adults, the broadly neutralizing antibodies actually undergo iterative rounds of the somatic hypermutation and a fitting maturation over several years before they're able to be potent and are able to neutralize a breadth of different kinds of combinations. But when we look at it in the infants, because there's a potential to elicit these broadly neutralizing antibodies without several iterations and without those mutations, this may be a potential key for us to bypass those several iterations and then potentially try to take that model and then the way that it binds, especially with this N332 glycan site, as a way for creating a template for vaccines and other treatments moving forward once we kind of establish the significance of this interaction. Oops. So in terms of where we go from here, it's important to remember that the immune responses to bacterial and viral infections differ really greatly between adults and infants. And logically, this does make sense because during pregnancy, the fetus itself is in a very low pathogen environment. And essentially has to have these regulatory immune processes because you're balancing both kind of the immune regulation of the mother and then of the fetus. And then when the child itself is born and exposed to all the gross things in this world, when it's a very high pathogenic environment, the immune system is very adaptive, which is again kind of underscoring the fact that this is a very appealing and an opportune time to kind of exploit and look at the dynamic shifts in the infant immune response itself. So when we want to look at the actual implications of what this could mean, is that because we know that the relationship between immunity and development between mother and child are similar. This could also be an important way for us to see, is there a link here between developing this immune response through active immunity between mother and child by studying that interaction. This is a controversial one, but obviously we don't have a lot of pediatric vaccine trials going on. The question kind of is, if we're not actually studying or looking for it in the population, how are we going to know how effective it is? So moving forward, it also would kind of be within our best interest or the interest of science to see how these actual vaccine candidates can work in infants. And the other important aspect of this is to identify the early markers of vaccine immunogenicity. So kind of being able to find these markers that will tell us that it's working, that we're getting an actual immune response, and it'll be long lasting. Figuring out what that looks like in the early stages to see how it plays out is going to be really critical when we start to look into clinical trials at these things. And in terms of the other developments that are being made at the moment, apart from these broadly neutralizing antibodies, that seems to be one of the hot topics in pediatric HIV vaccine development right now. People are currently looking at the use of the GP120 subunit, especially kind of looking at it in the context of these DNA vaccines. They've even also looked at the mRNA vaccine, but I know that a lot of the literature right now is saying that because we're kind of seeing the skew toward the humoral response in children, so again, humoral versus this cell-mediated T-cell response, they've been focusing a lot of kind of their energy and resources in this area of the field on looking at the B-cell lineage as a future step. And this is also important because the tolerance regulation of the B cells is significantly different in infants than it is in children. So this could also be one of the key reasons as to why are we seeing a difference, or why we're not seeing these specific therapeutic solutions not work in adults, but there's potential for it to work with a more dynamic, less developed, more plastic immune system. And just to kind of end off, the only way that we can actually properly and justly approach this HIV/AIDS epidemic is through a rights-based approach to health, in which every human has and is recognized their fundamental right to health. So this is both with respect to the current state of the epidemic, in which we do not currently have an actually ready to distribute vaccine, as well as when the time does come when we do have these therapeutics available, how are we going to get them out to people in a way that is just and people have equal opportunity to it. And just to kind of show a couple of pictures here, these were actually taken during my time in Cambodia. So obviously you can see it's not the most high resource situation, but having an appreciation and understanding for knowing that when you want to make a positive change, it's not always just a matter of having the science to back it, but also the political will to implement the advances you want to make. That's essentially the only way in which, across any and all strategies that we want to apply, that we'd actually be able to see an end, or have hope in seeing a potential end to the HIV/AIDS epidemic. So yeah, that's it for my presentation. That was a lot of rambling and confusion and things, but I hope you learned even something. Thanks.